Hello, everybody. My name is Luke. Like Moses said, I'm going to be talking about machine learning pipelines for biological applications. And so the way that we're going to talk about this, we're going to split up the machine learning pipeline into four categories: task definition, data collection, modeling and training, and then evaluation and interpretation. Likely there will be quite a bit of overlap between this talk and some of the other talks today. My goal is to give kind of a high-level overview from a statistical and data science perspective of some of the main concepts that we need to keep in mind when we're approaching machine learning pipelines, either from a kind of basic statistical perspective up to a like AI or deep learning perspective. These concepts will be applicable. It's also, these are going to be concepts that are applicable across a wide variety of domains, both within biological applications and elsewhere. We'll spend some time talking about model selection and evaluation strategies, and we'll also spend a little bit of time talking about some of the interpretability issues and communicating results. So it's typical to want our machine learning pipelines to be linear. So this is kind of the expectation slide, is that you define your task, you go out and collect data either by running experiments or by finding data online. You fit a model and you analyze the results and you're done. Oftentimes the reality looks something like this, where it's essentially you define a task, you collect some data, you realize that maybe some of the data was collected inappropriately only after you fit a model. And so you have to go back and collect new data, which leads to a new task, subtask. And this is just the reality of how science typically works. But the goal of my talk today is to move us from a pipeline that looks like this back to a pipeline that basically looks like this, the four steps in a more linear fashion. And we can avoid a lot of that kind of spaghetti pipeline by being careful with how we define our tasks, how we collect our data, how we fit our models, and so on. So we'll just kind of walk through each four of these categories step by step. So the first one, defining our task. Questions like how much data do I need? What kind of data do I need? What model is appropriate for my data? All are going to be contingent on the specific task that we want to tackle. And the success of our entire pipeline is going to be contingent on clearly understanding what we want to accomplish. So within machine learning and statistical applications, there's a huge number of both general models, like something like linear regression, or very specific models that are tailored to a specific application or a specific need. And it's quite overwhelming to even start with picking which method works for me, even after a lit review. So it's important to have as much clarity as possible about the goal of the research going into the data science component of any of this. So this slide shows a few examples. They're somewhat outdated, but many of these methods are still used, especially the kind of the statistical. Yeah, they're all still used, regardless. And the point is, how do I go about selecting which one of these methods is going to be right for my application? It's helpful to kind of work through some questions on the front end. So typical questions that kind of help frame our problem is my task going to be supervised or unsupervised? Supervised meaning I have some known label or some known response value that I'm interested in predicting or analyzing. Unsupervised meaning I'm maybe collecting information or collecting data on some samples, and all I can really do is cluster the data. I don't really have any notion of ground truth, whether the sample is sick or not sick or something like this. A second question, typically we want to be thinking about is my task going to be a regression task or a classification task? In other words, am I trying to measure some continuous value like height or something like this, or am I trying to classify values into sick, not sick? Is there a causal component? So this would be, do I expect certain predictor variables to be causing a response of interest, or am I only interested in looking at relationships and correlations? Hypothesis generation versus hypothesis testing. So these are cases where I might be interested in just exploring my data in general and coming up with new ideas for which to develop a study in the future. That would be hypothesis generation. Hypothesis testing would be, I have an established hypothesis. I think that something works a specific way. I'm going to develop a statistical model to confirm or deny that hypothesis. Single modality or versus multimodality. Will my task definition involve collecting data from different sources, essentially, different data streams that might not be aligned in space or time? And then lastly, what's kind of the ultimate end goal? Is this a research product? I'm writing a paper. It's kind of a static one-time analysis, or is this an applied application? Is it interactive? Does it need to be like web scraping data regularly? These are going to affect all sorts of model selection and downstream activities if model speed is going to be important and so on. So some kind of important questions. I know we kind of just went over some important questions when defining tasks, but I guess it's extended. These are the ideas, helpful things to be thinking about on the front end before you start collecting data and doing stuff with it. One is, how can I formulate my research task into an array or table? Sometimes this is a very obvious thing. Excuse me, Moses just messaged me and asked to let everybody know that he's lost his internet. So if there's nobody monitoring the Discord or the chat, let me know, I guess. I hear a lot of beeping, but I'm not paying attention to either of those screens. So
thank you, Luke. We can handle that on our end. Thanks for letting us know.
Okay, thanks. Yeah, sorry about that. So wanting to formulate the research task into an array or table. The gold standard tends to be that we can take whatever sort of research question we have and put observations or samples on the rows, and then variables, measurements as columns, or swap the rows and columns. But being able to actually define these things, and furthermore, subselect our features, those characteristics or conditions that our sample is exposed to, into responses and predictors. So we wouldn't have a response variable if we were doing an unsupervised task. But if we have a supervised task, then we have variables of interest that we're interested in maybe predicting or discriminating between somehow, and then predictor variables, which is basically anything that we think will affect that response variable or be helpful, useful for describing the response variable, or maybe anything that we're interested in being able to say anything about that relationship, whether or not we think there will be an effect or not. Other types of questions that are helpful at this stage in the pipeline is generally just asking, what level of interpretability is needed? Do I need to be able to know exactly not only what the relationships between variables are, but also how exactly the model is constructing those relationships? Sometimes this is required for certain standards, or something like drug development. You're going to have trouble extending deeply into deep learning without some guardrails on this interpretability, for instance. Is there a spatial or temporal component? This is going to be huge in data collection, because on the front end, if we can collect equally spaced measurements, our life will be much, much easier when we get into the modeling stage. And then lastly, having some notion of the cost of collecting observations for our task. If the cost is very high, we might be concerned about immediately thinking about machine learning or deep learning for our project if this is an experimental collection task. And we also want to be thinking about, what if something gets messed up along the way? Do I have enough bandwidth to recollect observations, or to maybe collect observations that I missed some subset of the population that I'm interested in the future? Essentially, expecting the unexpected. Can I plan for failure, plan for success by planning for failure? So an example might be changing a part out on the instrument between runs. That might be something that wasn't an original planned variable, but needs to be a variable in the final model, or you'll miss some important distinction between data that came off the instrument early and data that came off the instrument later. Another key consideration is that using a single response makes life easier. Most models are better fit for a single response. But you can save a lot of resources by collecting data from multiple responses and doing multivariate analyses. So just thinking through that ahead of time. And that leads into the third point, which is if you're working with a data scientist, this is the point typically to engage them. You don't want to wait until after your data is collected to engage data science. You want this to be collaborative from the very beginning to avoid some headaches. Okay, so we've talked some about task definition. Now we'll talk about principles for data collection. We need our data collection to be of sufficient quality and quantity. So it needs to be high quality data and also needs to be enough for drawing the model conclusions that we need to draw based on our task definition. We're going to have to format and pre-process raw data typically to get it into the format we need it. And decisions have to be made at this step about training and testing, cross-validation, these sorts of things prior to modeling. So first of all, data collection, thinking about sampling and representation. So one key thought at this stage, whether you're collecting data off an instrument or doing the actual data collection yourself, or looking for pre-existing data sets to use for a novel analysis, it's important to recognize that and unless I take a census or study every possible combination of every possible thing, my sample will not perfectly represent my population. That's the whole point of statistics, data science, and machine learning is to make inference from a sample about a population. I guess that's not the whole point, but that's generally the target that we're going for. We want to make inference from the sample about the population. And so in the top right, this is an example of chemical space coverage of a few different online databases. And the point here is that different databases are highly saturated for different spaces of interest. So if you're interested in a space that's not highly saturated, maybe it's not the right database for you. How much data do I need? This is a very challenging question, and it all depends on your research task and the model at hand. Typically, if you move from statistical methods like linear regression down to deep learning, neural networks, and so on, the data that you need increases. This is kind of a obvious thing to say maybe, but defining how much data you need, principles from statistical design of experiments can help with this. Also, literature review is a helpful place to go. You know, what have other successful research projects done? How much have they collected? And kind of using that as a benchmark. These are examples in the bottom left of some kind of traditional statistical design of experiments where the axes of those cubes represent different features that you might be interested in collecting over, maybe height, weight, temperature. I'm not sure what the specific application would be, but then the points on the cubes correspond to different levels of those features. So high and low, medium. And so defining all of these things ahead of time, if you're collecting data, is very helpful to say, you know, what's my minimum and maximum values that I might be interested in across all these features, and how can I iteratively collect samples so that I can narrow down what features I might be interested in. And then lastly, asking, is this a big data task? So am I going to have health records, mobile sensors, unstructured data? So that would be things like text, real-time streaming. All of these sorts of questions, kind of, I would say, non-traditional statistical questions are going to require considerations about data storage, data accessibility, alignment across variables, and so on, that are going to be important to nail down prior to data collection. Data formatting, I mentioned this previously, and this is not always the case. And in many cases, it's impossible or challenging to do with the type of data you have. If you have some unstructured data like text data, it can be challenging to get it into this format. But the kind of gold standard format for data is complete numeric tables. So where either the rows or the columns correspond to observations or variables. And ideally, every value in that table is filled in. You don't have missing values. We'll talk some about that in the future. But this is kind of the reason I say this is the gold standard is that the vast majority of machine learning methods will work on this type of formatted data. If you don't have to deal with unique formatting or missing values and so on, you'll have opened yourself up to as much flexibility in the future. Okay, so some key considerations for data formatting, getting my data into a usable table prior to fitting the model. Thinking through, is there any missing data? And if so, how is it encoded? This might not be a consideration if you're collecting the data yourself, but if you're going and scraping data from online or from some other research article, this can be a severe challenge, is kind of sorting out what data are real, what data are not, especially in cases where maybe zeros are substituted for missing versus zeros are truly meaningful. When you have numeric values, the model is not going to know whether 999 or 0. It's going to assume that that's a true value, and that can really mess things up. I've mentioned this a couple times already, but if the data has a spatial or temporal component, are the values aligned? So if I have multiple sensors collecting data concurrently, are they collecting data at the same time step? Or maybe I'm interested in a grid of data points. Are they equally spaced over a grid of property, or are they kind of randomly spaced? Different methods are going to apply to these different cases. Typically, the more aligned and the more equally spaced you can get things, the easier it's going to be for yourself. And then lastly, just identifying if there are unexpected values in the data, characters that should be numeric, et cetera. We could spend hours just talking about approaches for exploratory data analysis, EDA, which is kind of methods for looking at your data prior to stepping into the modeling steps to look for anything that's suspicious and kind of identify some of these problems beforehand so that we don't end up in a spaghetti pipeline, so to speak. All right, we'll talk about training and testing splitting now. So we typically split the data into a train and a test set. The idea is that our models are going to learn the data, learn the distribution of the data, and if we want to be able to apply this from a sample onto a population, we want to hold out some of our data that kind of represents unobserved data in the population so that we can evaluate how will the model work in real life when it sees data that it had never seen before. The training data is typically further split into multiple folds. This is kind of your standard machine learning setup, is something like a k-fold cross-validation, where the k just stands in for an integer. So it could be 10 folds. You divide your training data into 10 sets, and then you fit across a suite of models 10 times, each time holding out one of those 10 sets as your validation set to select which is the best of this group of models or group of hyperparameters in my models. Things like the number of trees that I use or the layers in my neural network or so on. And then after all of that is done, after we've kind of selected our model and looped over learning algorithm with our training data, then we have a final model that we select that we feed the test data into, make our predictions, and evaluate on that test data. One key consideration with this is that we want our training and testing sets to be equally representative. So say we have a data set that has 90 sick individual or 90 healthy individuals and 10 sick individuals. Our goal is to discriminate between healthy and sick. We want our training and testing sets to also have 90% healthy and 10% sick individuals in them respectively, even if our train set is only 20 individuals. So I guess in that case, it would be the test set would be, if it was 20% of the individuals, the test set would have 2 sick individuals. And also, we want to avoid data leakage. This is any time that the model is trained and it sees data from the test set. One of the most common ways that this happens is if we normalize the data using the test set. We normalize the training data using the mean or standard deviation or something that comes from the test set data. All right, so we talked about data splitting. We'll also talk some about data pre-processing and normalization. It's common for the input data to cover vast ranges, magnitude ranges. So we could have maybe age as one of our features, and then biomolecule abundance as another feature. And the minimum and maximum of those values can be extremely different. So what we want to do typically is have some sort of rescaling or normalization to get the features onto mean 0, standard deviation of 1, similar ranges. Some models are basically invariant to this, and it doesn't affect them, but models that have some sort of gradient descent component to them will benefit greatly. So neural networks will benefit greatly from rescaling ahead of time, and prevents issues of large magnitude inputs dominating in your early training. You want to have kind of a fair assessment of all of your inputs. Typical components of data preprocessing. So we just talked about this normalization. We also oftentimes will have to deal with missing values with either imputation or removal. Imputation means adding in a value synthetically when the value was missing. This can be done by borrowing information from nearby values or similar values, or just imputing a mean. We can also drop observations that are missing a lot of values. Another pre-processing step that's common is removing outliers. If the goal of our machine learning pipeline is to take a sample and make an inference about the population, if the outlier is not something that we would expect to actually exist in the population, in other words, it's something that was messed up in the data collection, we don't want to include that in our model. If we have a population that's very skewed, and that we expect there to be kind of these outlying observations, then we actually want to include that outlier in our model. It just depends on what's the nature of this outlier. And lastly, it's common to do feature engineering. So take categorical features. In this example, it's helix, sheet, and coil. I don't recall what those correspond to, but 3 categories. They can be encoded into ones and zeros, 3 columns of ones and zeros. Most models, depending on what software you use, most models will rely on some sort of encoding like this, either explicitly that you have to feed it in, manually encode yourself, or they'll do that under the hood. And then in the top right, this is an example from the PMAR-R pipeline, which is multi-omics analysis package, just kind of showing, yeah, these are kind of the steps that are applied in many different biological applications. We have to format our data, update missing values, drop features, drop samples, apply some normalization scheme. So the point of that is that different domains, different subdomains will have different norms or expectations about how these things should be done. So again, specific knowledge of your specific domain is going to be crucial here to, what kind of normalization should I do? What is an outlier or not, so on. So, okay, key considerations for this data preprocessing step: protect your raw data. So one is storage redundancy. We never want to overwrite any of our raw data. So wherever our raw data is saved, lock that down, create a copy, and then don't mess with the copy either. So never edit the raw data. I put that in there 2 times. It is one of those seeming very obvious things, but it does come up. And that's not a situation you want to find yourself in. Automating everything with scripts. This is common and easy to do nowadays, but using some sort of programming language and having scripts that go take your raw data into a processed or wrangled format before scripts that apply whatever modeling technique you want to do. If there's any sort of random generation in your pipeline at all, setting seeds for reproducibility is important. And then lastly, some of this exploratory data analysis. Plotting the data, looking for outliers. Does this data look like I expect it to look? You can save yourself a lot of time at this step in the pipeline, this data collection step, by thoroughly looking at your data and going through it prior to the modeling stage, in case you need to maybe recollect or collect some additional observations and so on. All right, so we're getting to the modeling, model fitting and model training. So at this point, if you've done a good job defining your task and collecting your data appropriately, the subset of models that you can pick from should essentially be predetermined, whether you have response variables or not, whether you're doing classification or regression. And our model selection should typically balance performance with performance on one end. So how good is your model at predicting the thing you want to predict? Balance that with, on the other end, interpretability, simplicity, and speed. We typically want highly interpretable, simpler is better, and faster is better, all things held equal. So machine learning modeling is about modeling observed behavior given your data. And the model that we select is going to depend on data type, quality, and quantity of the data. So this is, I think, a helpful flow chart kind of showing, okay, I'm at my model selection step. What kind of statistical or machine learning model is going to be most appropriate in my setting? It's important to note that these red boxes contain within themselves a large number of models. So, regression methods could include statistical methods like linear regression. It could include kind of traditional machine learning methods like random forests, regression random forests, and so on. But I find this kind of helpful to start thinking. I'll also say that this sort of decision should be made at the very beginning of the pipeline. Maybe not which specific model is going to be the final model you use, but the family of models, or maybe the list of models that you intend to evaluate should be selected at the beginning. Okay, interpretability. Interpretability refers to the ease with which the user can understand the model. So one notion of interpretability is which features or observations contribute to predictions. So this could be something like importance plots or Shapley or Lime or Ale or something like this. What's the relationship between my features and my predictions? Another facet of interpretability is, are the model rules simple to visualize or comprehend? In other words, can I understand, can I look at the model and understand how it got where it got? This is very easy with something like linear regression. I know that a 1 unit increase in this variable corresponds to an X unit increase in my response variable, or so on. And oftentimes we have this trade-off. So this is kind of a notional plot, but the deeper you go into deep learning, typically the better predictive power you get and the lower interpretability you get. So one principle to start off with is start with your most interpretable with your most interpretable models and your simplest models. And if they can solve the problem for you, then stop. Start with something simple and understandable, and move. Increase the model performance and the complexity of the model as needed. When we're training models, we fit the models on our training data, typically using some sort of cross-validation method, using folds in our data to avoid overfitting. So what is overfitting? I think this was talked about. I saw in the Discord chat some discussion about this already. So we might just be getting a double dip on some of this stuff. But the idea is deep learning models especially will perfectly learn the data that you hand to them, and can have a perfect score on the data set that you give to them. And so that's demonstrated here on the right-hand side, an overfit, where the actual shape of the data points we have, the blue lines, is not described by that squiggly red line. It's actually described as something more smooth. There's some inherent noise in that data that we want to generalize that we don't actually think is present in the true distribution. And so the point here is getting a perfect score on your training data is not necessarily something that you want to see. This is a indication that your model has memorized the training data and won't be able to generalize. We're typically thinking about hyperparameter tuning at this step, training models, not only comparing across different models, but comparing within a model. What happens when I look at a range of different combinations of hyperparameters? Hyperparameters are just, you can think of them as knobs that you tune underneath the hood of any machine learning model. So for a random forest, it might be the depth of the trees and the number of trees used. So we want to compare over kind of a grid or a collection of different combinations of these. Lastly, a term that you might be familiar with or might come up is ensemble methods. So it's common to fit a suite of methods and then average the results or take the most common prediction from those results. This tends to be a good way to leverage the strengths and avoid some of the specific weaknesses of a set of models. So kind of some last tips for model fitting here. Quarantine your test data just like you quarantine your raw data. Also, don't let anything see your test data. Leave your test data alone. Automate everything with scripts and set seeds. Use version control. Use Git so that you have a record of what you've done. Look at the data along the way with plots. Am I modeling what I think I'm modeling? Are my results making sense? And make sure you have both enough and the right type of data for the model you use. The best way to do this is checking the literature, also talking to statisticians, data scientists, other subject matter experts. And then lastly, beware of this underfitting and overfitting trade-off. These are a couple memes, I guess, to capture the goal of getting the right amount of fit. All right, so we come to our last component of our pipeline, which is evaluating and communicating our results. So like the modeling and training, the set of models that we use should be predetermined at this point in our pipeline. Similarly, the set of evaluation metrics should typically be predetermined. Maybe there are a few metrics that you're comparing, but at least the family of metrics will be predetermined. And interpretation and communication should always prioritize simplicity and clarity. So is it easy to understand and is it accurate and correct? So first of all, we'll talk a little bit about evaluation metrics. So it depends on the problem formation. One evaluation metric might be completely inappropriate for the specific type of task. This is especially true in cases where you have maybe a rare occurrence of an event. So you're modeling a rare disease or something like this. You're using accuracy, just the total proportion of correct predictions. You might have very, very high accuracy, but if you miss all of the cases of the rare disease, there are only a few of them in the first place, your model is very bad. And it's equivalent to if you just labeled everything as not having a disease. So selecting the metric is real. All right. So evaluation metrics can be very misleading based on our data or problem formulation. All right. So we'll talk through a few common evaluation metrics just to give some overview of these. And many of these will probably be familiar, but it's helpful just to talk through the basics again. So if for classification tasks where I may be looking at two different labels, sick versus not sick, common metrics used, the first one is accuracy, the percent of correct predictions. Precision is the percentage of correct positive predictions. So in this plot over here, it's a little confusing. The blue side corresponds to everything. Say that negative means you're not sick, and positive means that you are sick. So everything over here with the hollow points are observations or samples that were not sick. Everything with the solid points are observations that were sick. And then everything within the green is what has been classified by the model as sick. So the subset over here in the left side of the circle with solid points are correctly classified sick subjects. And then the observations outside of the circle with hollow points are the correctly classified not sick or healthy samples. So precision is the percentage of correct positive predictions. Recall is the percentage of all of the sick samples that were correctly predicted as sick. So you can see that depending on whether you have maybe a rare occurrence or what the cost of a true positive versus a false negative is, or false positive versus false negative, there's a trade-off there depending on your context. What might be more costly? You might want to pick the metric that captures that cost best. Other metrics, FF score, F1, this is essentially the average of precision and recall. So trying to find a balance between those two. And then area under the ROC curve. This is the proportion of true positives or Yeah, it's the sensitivity versus 1 minus the specificity. And essentially, a model that has a curve that's closer, the area under the curve closer to 1, um, the model performs better. So just a few examples of how metrics can be misleading. So a model that chooses true 100% of the time has 100% recall. So using recall alone is not sufficient. A model that gets 1 right out of 1,000 can still have 100% precision, depending on the setup of the labels. And so that's why something like an F score can be useful to kind of take the average between those two. And then lastly, this is the example I was describing before. If you have 99 images corresponding to healthy and 1 corresponding to disease, even a poor classifier will achieve almost perfect accuracy. So the point is you'll need more than one metric typically. So similarly, regression metrics, this is just a few. There are many more. Commonly used regression metrics are mean absolute error and root mean squared error. These are both measuring a very similar value. It's essentially the difference between the observed and the predicted value. With root mean squared error, because it's squared, larger errors are tended to kind of be given more weight. So depending on whether you want to penalize large errors or not. And then R squared is another metric, which is looking at the improvement of the model over against simply using the average as a predictor. So when your R square is closer to 1, it's better. Okay, so key considerations for model evaluation. So just as a kind of recap, because I don't think I included this in the previous slides, your training and evaluation workflow is to select evaluation metrics, train over your suite of models, as well as hyperparameters within models using the best metric, with best metric performance using the training data, basically. And then apply the selected model to your testing data and evaluate the same metrics performance. So select your metrics carefully. Accuracy is not always the best choice. It's typical to select multiple metrics and either look at an average or just look at all of them and make a somewhat qualitative evaluation on the quantitative metrics. And then lastly, beware misleading metrics. So silly example, but if you have a database of Gatorade drink colors and a sample of Windex, it will be identified confidently as blue Gatorade. This is a big problem if you plan to drink it. All right. So then lastly, a couple slides, one on interpretation and one on communication. So some machine learning methods have built-in interpretability tools. So this is something like random forest has importance plots. That should be importance, not important. Sorry about that. Other models might not have a built-in interpretability tool, but tools can still be applied kind of post hoc. So Shapley values are an example of this. ICE plots or ALE plots are also examples of this. And what these sorts of plots do is they tell us something about the relationship between the features and the responses, or the predictor variables and the responses, both what is the directionality, and the importance. Does it appear that one feature is more important than others? Big challenges with interpretation involve correlated features. So oftentimes machine learning methods might average out two features that are highly correlated, so that if you had only selected one of them, it would be important, but because you have both of them, neither of them are very important. This is also an issue with statistical models. And oftentimes these interpretability metrics are looked at kind of marginally. So that means just looking at one of the features at a time and then plotting them together, as opposed to looking at the combined effect. Shapley values are trying to get away from that kind of marginal look, but there's still some challenges there with correlated features. So one of the best things to do is have features that are perfectly independent, which is rarely possible, but that's kind of the goal oftentimes of experimental design. Key questions at this stage is, does the model's interpretation make sense in my specific context? And then does the model suggest unexpected results that might suggest errors in the data or the training method? So this is one of those, back to that spaghetti pipeline plot. This is a case where before going and writing results, if the interpretability just makes no sense, as the subjects increased smoking, they got healthier, or something like this. You want to go back and double check that that those results are true and not just because you accidentally swapped the labels somewhere along the way. All right, last slide, communicating results. So three things. One is data and model reporting. Okay, we're writing up our report. How do we include information about the data we used and the models we fitted and reproducibility? So one, this is kind of this idea of fair data. So it's becoming common to require data to be findable, accessible, interoperable, and reusable. The idea is, we want to be able to access this data and reapply the same experiment that you ran and get the same results or close to the same results that you got. Secondly, it's common to put together data cards or model cards, which basically capture, how was this data collected? What were the different packages used? What was the training environment, the parameters and the weights, so that it can be just as clear as possible, all of the different steps that went into things for the sake of reproducibility. A note on visualization. I really like this quote by Edward Tufte. Excellence in statistical graphics consists of complex ideas communicated with clarity, precision, and efficiency. So some notes on visualization is keeping your results understandable. So large legends, large axes that are readable, titles that are clear, accessible colors. So like colorblind accessibility, being accurate, so avoiding misleading axes. This can be common. Maybe if you're plotting a grid of plots, of many panels of plots, you want the axes to the X and Y axes to be the same typically across all of those plots. And then lastly, reducing clutter. So you shouldn't have one piece of information corresponding to multiple dimensions. You're plotting sick versus healthy. Sick is colored red, healthy is colored green, and sick is an X shape and healthy is a circle shape. You can pick either the color or the shape. You don't need to include color and shape. This is cluttering and confusing because it requires, I have to now look at the legend and keep one more dimension in my mind as I'm interpreting this plot. Lastly, here are some kind of common tools in R, Python for reproducibility that are worth getting familiar with if we're going to be doing work in the machine learning world for biological applications. And then lastly, just a plug for using GitHub or some GitLab or some other form of version control. It can be painful to learn it at first, but the reward is high when you need to go back in time and back to the spaghetti plot or the spaghetti pipeline, have to go back and restart something. It's very helpful to have a history tracked for you automatically. This is just one last quote. This is probably familiar to some of you, many of you maybe. 70% of researchers have failed to reproduce another scientist's experiments, and over 50% have failed to reproduce their own experiments. Now, this was a study done about a decade ago, and I think that this number is inflated compared to I think this was across, not inflated, sorry, not accurate for the domain of biology. So I think that different domains, it looked at a lot of different academic disciplines, and different domains were worse offenders. But the point remains that we want to be hyper vigilant about reproducibility and can I even redo the experiments that I ran and get the same results?